All right, so this is the extraction lab. So this is where you actually break down a piece of plant tissue and extract usable DNA from it. And how do you do this?
You just use a big grinder, like a coffee grinder, to grind up all the dry leaf tissue. And then you incubate it, with enzymes so it breaks up all the cells. You get the DNA out.
And is that a problem for things like cypresses where they have a big waxy cuticle or where there's a...
Oh, yeah, but you figure it out. You figure out what you have to do.
How do you know if you've got the actual tissue and you've gotten through the cuticle? You have to...
You just grind it up real good and incubate it overnight.
And that cuticle won't matter as long as you grind it up well enough. And,
yeah, dry it well enough. I mean, some things are hard, like succulents, but you figure it out. Comps are not that bad. And then this is the filter that I was telling you about. This is called the Lilac Shredder. Comes with this, kit, the Kyogen kit for extracting DNA, which is kind of like the easy way to do it.
Right.
There's a filter in there. You load, your material in there, and then you put it in a centrifuge. Certain things can pass through the filters, and others can't.
And they'll pass through the filter when the centrifuge spins them.
Yeah.
Basically.
So you do this a bunch of times, and then you get some purified DNA.
And so once you have that, then what do you do? Then that's when you put it in the PCR machine?
Well, first you use some primers to amplify certain parts of the DNA.
Yeah. Which is either ITS or chloroplast or...
Yeah, here, look at the ITS thing. Here's the diagram. So this is the ribosomal DNA. And you've got the 18S here and the 26S nuclear DNA. These are just super conserved parts of the DNA molecule because they code for, really
Where's the variable parts
machinery.
Are right here?
Yeah, these are spacers in between like the very well conserved parts of DNA. The internal transcribed spacer is what it's called, ITS.
Right. And so, but for most things, these are going to be the same. But this is where you get the variation that can help you determine what species or how closely related things are?
Yeah, you can figure out relationships between things, or you can figure out divergence, whether something's a new species or not. And so what you do is you use a primer, like ITS Lu, that's the complement of this last little part of the 18S, and you use another primer, like, ITS4 or ITS2, that's the complement of this last, first little bit of the 5.8S. And then what you get is what's between the two, ITS. So you amplify
a forward
that.
and a reverse primer, right? And then another one here.
Yeah. So you amplify that region of DNA using PCR, and then you sequence it.
This is the most common one. When do they use chloroplast DNA?
When?
Yeah, like in what situation would necessitate the use of cpDNA?
Well, ITS is useful for animals, for fungi, and for plants, but chloroplast is just useful for plants.
Right, but we're talking about plants. They still mostly use this, right? When would they use chloroplast? What would determine that?
Well, ITS is useful for deeper relationships because it's super well conserved.
Species?
Yeah,
you mean?
It would be useful for species. If you wanted to resolve anything below that level, or even in the cases where ITS isn't variable enough to figure out the species, like in bulbs, because geophytes are perennial, and they're long-lived, then you would use chloroplast DNA, and that's more variable.
So chloroplast is going to be better for lower taxonomic levels than species, like subspecies and varieties and shit like that, or even different populations within a species?
Yeah, and also the evolution of the chloroplast is completely different from the nucleus.
Right.
It's a completely separate part of DNA, and it's uniparentally inherited.
Right. I'm just talking about for molecular phylogenetics.
Personally, the chloroplast can also be tricky. Because pollen, because the chloroplast has a much more complicated history of jumping around, and plants hybridize. So you end up with, results that don't make any sense.
Yeah.
For the thermocycler that have been tested over the generations.
What does that mean, programs for the thermocycler?
You tell the thermocycler to heat up to 96 degrees for 10 seconds, then 60 degrees for 30 seconds, 72 degrees for 20 seconds, and then it repeats that 40 times.
And during that process, the DNA is unzipping and bonding with the primers?
Yep, bonding with the primers and copying, and then unzipping. You start with 1, and then you get 2. Then with 2, you get 4. And so on and so on. So it's exponential.
It's 96 degrees Celsius, yeah.
Yeah.
And what's the enzyme that comes from the extremophile bacteria, from the thermophilus?
So you're using the Taq polymerase from an extremophile bacteria, right? From hot springs, somewhere,
Right.
maybe in Yellowstone or something
Right.
like that. And so they can withstand these really hot temperatures, and they can continue copying the DNA.
Uh-huh. What you do with the aid of this PCR machine? That's a fancy one. I've never seen one that looks so new.
I don't think it's that new. It's kind of from the 90s or something. Look at this.
No, it's better
Look
than
at the colors.
it's better than the one they got at Counterculture.
Check this one out.
What is the spore and fill? Who named these? Why do they have two names?
They're called sporophyll. Look at this one. This is Methuselah.
Oh, this is an old ass one, huh? But it does the same thing that those do?
Yeah, and it's kind of better too. It's an oldie but a goodie.
Real nice.
Look at from there.
Right, Leia discordia. It's a serpentine endemic from, what is it, south of Pinnacles?
Yeah, just south of Pinnacles. It's only known from a couple spots, serpentine outcrops.
How recent is it? Can they tell how recently it speciated?
Very recently, yeah. And it's one of these things that is endemic to a cooky kind of soil. But it's also discoid, and its most closest relatives they have rays. So it's an example of something moving onto a cooky soil, losing those discs and becoming a discoid.
What would be the
plant.
purpose of losing rays if you're an aster?
Well, you're growing on this super stressful
They
environment.
probably cost a lot to make, yeah.
Maybe. But I don't know. There's a thing with a lot of serpentine endemics. They tend to be more cryptic
Uh-huh.
because they're growing on this really stressful environment. If they lose their tissues to some bugs,
Right.
they're in trouble.
What's up with the 22,000 kilobase pair inversion in Asteraceae? All but the basal asters. What do you know about that? What can you tell me?
It sounds like you already know.
No, I don't know anything. I just read about it. I just read the paper by Funk.
Yeah, inversions are cool. They're probably way more common in plants than we think.
Nothing else has this except the non-basal asters.
Right. It's one of the ways that you know that those asters form a monophyletic group.
Right.
With more basal asters as an outgroup to them.
But why would a plant do that? Why would it get a 22,000 base pair mutation or kilobase pair mutation?
Inversions happen. One of the reasons inversions happen is because they can make it easier to have local adaptation. When you invert parts of the DNA, like in Mimulus guttatus, you could look this up. In Mimulus guttatus, there's an example of an inversion in those perennial. Well, now it's what? Diplicus.
Diplacus, right? Yeah.
So there's an example of an inversion in the perennial ones that's not present in the annual ones, and it allows them, a certain.
All
amount
perennial
of local
Diplacus share this
adaptation.
inversion, and none of the annuals do.
No, just guttatus.
Just guttatus. Oh, yeah.
So I don't know. I guess when people were studying the chloroplast DNA and really getting in there and getting into the dirt of it, discovering that inversion was really useful because it was a way to figure out phylogenetic relationships.
Right, that they all descended from this one common ancestor.
Yeah.
And it's just guttatus that has it?
No, lots of plants have those.
Well, no,
inversions.
but in this case, you're talking about this inversion as just present in guttatus.
Yeah.
How big is it?
I don't remember. You should look it up.
Is it a fucking big one like the aster inversion or what?
Yeah, it's a big one. It's on a chromosomal scale.
Yeah. God, that's a cool fucking genus too, man. I would love to learn some more about that, at least, molecularly. All right.
You want to go check out the herbarium?
Yeah, let's go. Okay, so you
So you put the...
grab this stuff out of the PCR,
Yeah.
right?
You do some centrifuging.
Wait, we're getting ahead of ourselves. You do this before the PCR.
Well, you got to just make sure all the liquid gets to the bottom, right? So you
Right.
centrifuge a little bit. And then this is what you want. You want to make one of these. You got to find out if the amplification was successful.
Which you do through electrophoresis.
You have a gel.
Mm-hmm.
It's like a jelly. And you put the DNA in a little well. And then you run an electrical current through. The DNA will move through the gel. And then you image it with UV light.
And that's because it's negatively charged going to positive, right? Because it's a phosphate? What is it?
Yeah.
Yeah.
Exactly. So these are the gels, right? This is the little boat that you fill with agar, and that you put your stain in there. You fill this with the liquid, and I can kind of show you. This is how you get the electric current running. Attach it. It's just like I don't know. I guess you don't really do much of this kind of stuff in everyday life.
Nah.
But you get an electrical current running through there. The DNA runs this way.
I've seen electrophoresis done before at counterculture, but
Yeah.
they don't have as nice a setup as this.
No, it's pretty nice. And then so you take that gel, you stain it, usually in some nasty kind of fluid.
Mm-hmm.
You got to be careful not to toxify yourself. And then image it with UV light, and that's how you can tell if the PCR was successful.
All right.
All right, so it's a little blurry, but each one of these wells here contains some DNA. You ran the current, and the DNA was traveling this way. So you can see in this one, the DNA went this far, and it ended up here. And then you imaged it, you get a little line. In this one, there's no line. So that one was unsuccessful. So you might have to go back to, your DNA.
So this is basically
PCR.
successful is there's a mark right there.
Exactly.
Unsuccessful is there's not.
Yeah.
And what is this over here exactly?
Same thing. I just ran a gel that had 2 wells. Each one of these was filled
Oh,
with
okay.
DNA.
So this is 2 separate. Okay, I see
Yeah.
what you're talking about.
And you can also use this. You can use this to measure the length of the DNA fragments that's in the sample. So,
Meaning measuring it in what, base pairs or
yeah,
what?
more or less. You use this is called a base pair ladder right here. It's like a standard,
Uh-huh.
and it's got fragments of DNA of different sizes. So smaller pieces are going to travel through the medium faster, and they're going to get farther. And bigger pieces are going to have more friction, and they'll end up closer on this side. So you can see all these fragments kind of fell out in the same area. They're all about the same size because ITS is a DNA fragment of a given length, and these are all
What is it, 1,500
amplifications.
base pairs or something like that?
No, it's shorter than that.
Really?
800 or so.
Oh, shit.